All right, let's just jump right in. Our DNA, it's basically this massive, massive instruction book for life. It's got everything in it that makes you, well, you. But have you ever stopped to think, how on earth do we actually read that book? Today, we're going to look at two amazing technologies that do exactly that, but in ways that are like polar opposites.
And that's the big question, right? We're talking about 3 billion letters. That's not a typo. For a long, long time, trying to read that was a monumental task, almost impossible, really. But then two key players came onto the scene and completely changed the game. So let's meet our first contender. So our story starts with the OG, the original gold standard. It's a method so precise, so elegant, you can really think of it as a master calligrapher.
And its name is Singer sequencing. This technique came about way back in 1977, and believe me, it was absolutely revolutionary. It's what gave us our very first glimpse into the human genome. And its whole philosophy, its core principle, is all about focus. It just meticulously reads one single piece of DNA at a time.
Yeah, to really get a feel for it, just picture that scribe, right? Carefully, deliberately writing out one single, perfect sentence with a quill and ink. It's not about speed at all. It's slow, it's methodical, but the result? It's pretty much flawless. It's all about getting that one sentence just right. And that's why it's called the gold standard. I mean, it is super accurate. Plus, it gives you what scientists call long reads. It can read a pretty long stretch of DNA, like up to 900 letters, in one shot. That's a huge deal, because it gives you a lot of context around that specific spot. So you can see why it's just perfect for jobs like verifying a single mutation you already think is there, or for sequencing small, manageable chunks of DNA.
Okay, but what if your goal isn't just one perfect sentence? What if you need to print the entire encyclopedia and you need it done by tomorrow? Well, for that job, you need a totally different machine. You need an industrial strength printing press.
And that, my friends, is next generation sequencing, or NGS for short. Now, the key phrase you gotta remember here is massively parallel. Instead of focusing on one little piece of DNA, NGS basically shatters the whole genome into millions of tiny fragments. And then, and this is the crazy part, it reads all of them at the exact same time.
I mean, the scale is just, it's hard to even wrap your head around. We're literally talking about millions of tiny DNA fragments all being read simultaneously. It wasn't just an improvement, it was a total paradigm shift in sheer volume. And all that volume, all that parallel processing, it gives you one incredible advantage: speed. Unbelievable speed. With NGS, you can sequence an entire human genome, yeah, all 3 billion of those letters, in about a day. Seriously, just let that sink in for a second. The very first human genome project that used Sanger sequencing, it took over a decade and cost billions of dollars. NGS comes along and compresses that whole monumental effort into about 24 hours, and for just a tiny fraction of the cost. So this wasn't just about making things faster. It completely democratized genomics. It took it from something only a few elite labs could do and put it into the hands of thousands of researchers all over the world.
Okay, so we've met our two players, the precise, meticulous calligrapher and the lightning-fast printing press. Let's put them head-to-head and see how they really compare.
And let's kick things off with maybe the biggest difference of all: money, cost. If you think about it as paying for every single letter of DNA you read, Singer gets pretty expensive fast. But with NGS, because you're reading millions and millions of letters all at once, the cost for each individual letter just plummets. It's incredibly low.
So let's break this chart down a bit. First up, throughput. That's just a fancy word for how much you can do at once. With Sanger, you're looking at one target. With NGS, you're looking at millions. It's not even a competition. Then there's discovery power. This is huge. Sanger is fantastic if you already know what you're looking for, right? To confirm a hunch. But NGS, it just casts this enormous net and can find genetic variations you never even knew existed. Next, sensitivity. Think of this as finding something really rare. Imagine you're looking for a mutation that's only in a few of your cells. Sanger might miss it completely, but NGS can dial down and find variants that are present in as little as 1% of a sample. That's a total game changer for things like cancer research. And finally, cost per sample. Now this is interesting. If you're just looking at 1 or 2 genes, Sanger is actually cheaper. But if you need to look at hundreds or thousands, NGS wins by a mile. That initial setup cost for NGS, what they call library prep, is higher. But after that, the cost per gene is just tiny. So you see, this really isn't about one being better than the other. That's the wrong way to look at it. It's about having two very different tools for two very different jobs. And the real art, the real skill, is knowing exactly which tool to pull out of the toolbox.
So how do you choose? Well, it all boils down to the question you're asking. What's your mission? Are you trying to confirm something you're pretty sure is there, or are you heading out into the great unknown on a journey of discovery? You're going to reach for Sanger when what you need is laser-focused, cost-effective precision. So, for example, say a doctor thinks a patient has a specific, well-known genetic mutation. Sanger is the perfect tool. You just go in, check that one spot, and get a super accurate yes or no answer. It's quick, it's cheap, and it does that one job perfectly. But you go with NGS when you are on a true mission of discovery. If you're out there hunting for a brand new virus, or trying to find those super rare cancer mutations that are hiding in a tumor, or sequencing someone's entire genome to solve a medical mystery, you're basically looking for a needle in a haystack. And for that job, you need the brute force, massive power of NGS. So as we start to wrap this all up, it becomes really clear, doesn't it? These two technologies, they're not rivals. Not at all. They're partners. And together, they really form the backbone of modern biology.
When you boil it all down, it's this simple. Sanger is for precision. NGS is for scale. One gives you the perfect close-up look. The other gives you the sprawling panoramic view. And you know what's really cool? Scientists use them together all the time. They'll use NGS to cast that wide net and discover something new, and then they'll use Sanger to zoom in and confirm it. They're the perfect team.
And that's a pretty powerful idea to end with, isn't it? Sanger sequencing first opened the door to that massive book of life, but it was NGS that put a key to that door in the hands of almost every researcher out there. The pace of discovery now is just accelerating like crazy. It really makes you wonder, what incredible secrets are we going to unlock next?